அதனால என்னோட தமிழ் ஒரு 3 வயசு பையன் மாத்திரம் தான். அதுக்கு பதிலாக நான் உங்களுக்கு கூட இங்கிலீஷில் பேசுகிறேன். So, first of all, I want to thank the organizers for arranging this, but I especially want to thank the publisher and the translator for bringing out a Tamil edition. And I can imagine that the translation was not straightforward, partly because it mixed a lot of very personal and idiomatic English, showing sort of personal humor and personal relations on the one side, and with highly technical science on the other side that has to be translated. And you heard about how he had to almost invent technical terms in Tamil. So thank you very much for doing that. So I want to talk to you about the 4 themes in Gene Machine. Scientists, when they often write memoirs or autobiography, by the way, this is not an autobiography. It doesn't talk about my childhood or parents or anything like that. It's really focused mainly on the science and on a particular, we have many thousands of them. So, for example, humans, this is an overestimate, I need to revise this slide, but humans have about 20 to 25,000 genes. And it's about the same number of genes as something which is a worm. This is a worm that has about 20,000 genes. And this Arabidopsis thaliana is a weed, and that grows sort of almost without limit. And it has about 20,000 genes. So we don't have more genes than a worm or a weed. So that should give us some perspective on where we stand in life. Now, as I told you, genes are units of information, and by and large, they contain information about making proteins. Not just how to make them, but also when to make them and how much to make them. So there's a lot of control there. But fundamentally, each gene codes for information to make proteins. There are also some genes which don't code for proteins. They're called non-coding genes. But I won't talk about that today. Now, if you look at proteins, they're long chains. They're like polymers with different types of chemical groups attached to them. So there are about 20 different types of building blocks called amino acids. And you can string them together in a particular order, and that's what a protein is. And you can represent them in the English alphabet. Since there are only 20 or so amino acids, you can represent them by single letters in the English alphabet. Now, if you look at what genes look like, what proteins look like, they can come in many different shapes. And I particularly want you to notice this one, because this structure of this protein, collagen, was discovered right here in Madras, because it was discovered by Venki Ramakrishnan at the university when he was working at the University of Madras, and it was a very famous discovery. And this is a protein called hemoglobin, which takes oxygen from our lungs through our blood to our tissues where it's needed. You're able to see the screen because of this protein, which sits in the membranes of the cells in your eyes, and it can detect light, and it's called rhodopsin. So this is just to show you proteins come in many different shapes and carry out many different functions, but they're all made using information in a gene that codes for that particular protein. So for each protein, there is a particular gene, and we have thousands of them. So how can that work? Now, DNA, as you know, is a double helix, and it has 4 types of building blocks called bases. So you can think of DNA as a sentence written in a 4-letter alphabet. Now, what happens is parts of DNA that code for a gene are copied into a temporary molecule called mRNA. Now, nobody knew about mRNA 3 years ago, except for scientists. But of course, everybody now knows about mRNA because of the COVID vaccines. the mRNA vaccines are some of the best vaccines against COVID-19. And what that mRNA is simply a stretch of RNA that codes for the spike protein of the coronavirus. And your body uses that mRNA to make the spike protein. That's how you make the immune response. And then from this mRNA, you have to go to make a protein. But remember, there are 4 types of bases here, but there are 20 types of amino acids here. So you're going from a 4-letter alphabet to a 20-letter alphabet. And the question is, how is that done? Well, what the cell does is it reads groups of 3 of these bases at one time, and each of those groups of 3 codes for an amino acid. So if you only had a 1-to-1, you'd only be able to make 4 amino acids. But if you had 2, you'd have 4 times 4, or 16, which is still not enough. But if you had read them 3 at a time, then you have plenty of redundancy to code for amino acids. Now this doesn't happen, and the way that happens is that there's a molecule called tRNA which recognizes these 3 bases on the mRNA and brings along the right amino acid. There's a different tRNA which recognizes the next group of 3 bases, brings in a different amino acid, and so on. Now, this doesn't happen by itself. Rather, when people were trying to find out where proteins were made in the cell, they found that the proteins localized in these blobs in the cell, and when they isolated these blobs, they found there were particles which all looked about the same, but had 2 parts. If you look, each one has a large and a small part. If you look at all of them, you'll see they consist of a large and a small subunit. These particles were called ribosomes, and that's the name we've used ever since. And the ribosomes are amazingly complex. So for example, this is the ribosomes from bacteria. They have about 50 proteins and very large pieces of RNA. It's about 2/3 RNA itself. And in humans, it's much bigger. There are about 80 proteins and very large pieces of RNA that make an even bigger ribosome. But they all consist of these 2 subunits. Now, many years, many decades of biochemistry led scientists to figure out that the small subunit binds mRNA, which has that genetic information. Remember, mRNA is a copy of a particular section of DNA which contains that genetic information. And then the ribosome has slots in it for the tRNA molecules. And it figures out where to put the first tRNA, and then it brings in the second tRNA with the second amino acid, and then it links the first and second amino acid and moves, and then a third tRNA can come in and deliver the third amino acid. So the ribosome is basically stitching together the amino acids to make the protein, but the amino acids are only from the tRNAs that correspond to the code. So it's reading the code and bringing in the appropriate tRNAs which bring in the appropriate amino acids. This is how proteins are made by the ribosome. So I want to switch to how I got into ribosomes. I said to you that throughout the book you'll see that there's a feeling of being an outsider. The first time I was an outsider was when I went from Chidambaram to Baroda at the age of 3. And I still remember my first memory of going to Baroda was standing at a playground and not understanding anything that anybody was saying. All the children were playing, and I couldn't understand a word they said, because at that time I only spoke Tamil. So the other feeling of an outsider is I went to a co-educational school, but when I was in 3rd grade, the nuns who ran the school converted it to a girls' school. So as I went on from 3rd to 4th to 5th, boys would leave, but no new boys were admitted. So when I was in this is I think 10th standard, you can see there are about about 5 or 6 boys scattered around near the edges in a class of girls. So I was a minority and outsider in my own school as well. So that was another interesting thing. So then, as you know, I graduated in physics from the University of Baroda, and at the age of 19, I went off to do a PhD in physics in Ohio. And this is where I spent 5 years of my life. And I like to joke that the only really productive thing I did in those 5 years was that I met and married my wife. So what was the problem? When I looked at physics, I had no trouble taking exams. I did fine in my coursework and exams in physics. But when I started doing research, I realized the problems in physics are very hard. I don't mean exam-type problems. I mean making real advances in physics seemed very hard because physics is quite a mature field. And there have been very few really big breakthroughs in the last few decades. And I didn't want to spend my life doing boring calculations that didn't amount to anything. But then when I looked at biology, I found that almost every issue of Scientific American had major breakthroughs. And many physicists had made this transition from physics to biology. And I thought, well, maybe I should switch to biology because it seems not only at a very exciting stage, but it also seemed like, ordinary people like myself could make a good contribution. Whereas I felt you had to be perhaps, exceptionally brilliant to make a breakthrough in physics. So anyway, I thought of doing a postdoc because I was already getting a PhD. But I didn't know any biology, so I decided to go to graduate school all over again in biology. And by that time, I had been married only a year earlier, and I had a stepdaughter who was 6 years old, and my son was born, and he was only 6 weeks old. So with these 2 children and my wife, I drove a truck with all our possessions from Ohio to San Diego, where I spent 2 years learning biology at this University of California in San Diego. And by the end of the 2 years, though, I had learned quite a lot of biology, including some very comical mistakes that you make when you don't know anything. And I'd taken undergraduate courses, even though I had a PhD. In my first year, I took undergraduate courses in biology, not just graduate courses, because I didn't know anything, and I had to start at the beginning. But at the end of 2 years, I felt I had learned enough biology to actually do lab work and to do research. And then I thought, maybe I don't need a second PhD, work another few years to get a second piece of paper saying I have a PhD. And at the time, I noticed an article from 2 professors at Yale on the ribosome using a physical technique. And one of them was somebody who had offered me a postdoctoral fellowship 2 years earlier, but I had declined, saying I'm not ready for a postdoc. But then I wrote to him and I said, look, you were interested in me 2 years ago when I didn't know anything, and maybe now that I've learned some biology, I might actually be more useful to you. And he wrote to me and he introduced me to his colleague, Peter Moore, and they offered me a postdoctoral fellowship. So 2 years after I moved from Ohio to San Diego, again, now my son was 2 years old, and we all packed up and moved again across the country to the East Coast, where I moved to Yale University. And at Yale University, I worked under this man, Peter Moore, who was actually Jim Watson's graduate student. And Watson is known for DNA, but his lab also pioneered a lot of the early work on ribosomes. So many of the early ribosome researchers came out of Jim Watson's lab, including my professor, Peter Moore. And we were looking to see how the proteins in the small subunit were arranged. And that's what I spent 3.5 years doing before I finally ended up getting a job. Initially, by the way, I applied for 50 job applications and didn't get a single interview. And in the end, I got a job that wasn't very satisfactory, and then I had to move a year later and so on. And that's all described in the book. But moving forward a few years, it struck me that, if you want to understand something in detail, you have to know its atomic structure. And I'll explain why. For example, if you didn't know anything about a car, and you didn't know what it looked like in detail, from far away it might just look like a dot that's moving around. Now, if you get closer, you start to see that it has a shape and it has lights, and you learn something about a car. And maybe you learn that it consumes gasoline or nowadays electricity, and generates power and generates pollutants, carbon dioxide and so on. Now, if you look at a car in more detail, you start to see it has 4 wheels and an engine that maybe delivers power, and maybe a steering wheel that allows it to go in a certain direction. But you still wouldn't understand a car. But if you look at a car in great detail, and you understand how everything is put together, how the engine has pistons that move up and down, and there's an ignition that fires a fuel mixture, and the power generated by the piston is transmitted to a crank which turns the wheels, then you start to understand how a car actually works. So you have to be able to visualize things in sufficient detail in order to understand how things work. And the same is true for molecules. In fact, even from the very first structure of a molecule, which was sodium chloride, we actually learned something from the structure. Now, molecules, the ribosome is a very large complex. It's, 500,000 atoms. But it's very small by everyday standards. And the question is, how do you visualize detail in something that is very small? So normally, if you have an object, you would put a lens in front of it. And what the lens would do is collect scattered rays from the object and recombine them into an image. In fact, all of you are doing that now through the lens in your eyes. It's combining scattered rays from the object and recombining them into an image on your retina. Now, if the image is magnified, then of course you can see more detail, and that's how a microscope works. But if you have a molecule, the problem with molecules is they're too small even to be seen by a light microscope because of a law in physics which has to do with the wavelength of light. The wavelength of light is much larger than the distance between the atoms in a molecule. So in order to determine the structure of molecules, a classical technique was X-ray crystallography, where you persuade the molecules to form 3-dimensional stacks, which is to say very regular 3-dimensional arrangements, which is what a crystal is. And you then take the crystal and hit it with an X-ray beam, and the crystal then scatters the X-rays. Now, what you can do is you can recombine these scattered X-rays in a computer, which is essentially what a lens is doing, is recombining the scattered rays. But since there's no good lens for X-rays, you effectively measure the scattered rays with a detector and recombine them in a computer. And when you do that, you get effectively a 3-dimensional image of the molecule. And that's how structures were determined. Now, I didn't know anything about this technique called X-ray crystallography. So I went away on a 1-year leave of absence, a sabbatical, from the United States to Cambridge, which is where this technique had been developed in the 1950s. And the director at the time was a very famous man, Aaron Klug, who was a Nobel laureate at the time, and he later went on to become president of the Royal Society. Now, when I wrote to him, I was not a particularly known scientist. A few people in my field knew me, But luckily, he didn't throw my letter into the waste paper basket. But he knew about my work, and so he liked me enough to sponsor me for a sabbatical. And sponsored me for a Guggenheim Fellowship, which is quite a prestigious fellowship, often given to authors and writers and others. Anyway, so I went there to learn crystallography. And when I came back, I thought, well, maybe it's possible to do the structure of something even very large, like the ribosome. Now, this person, Ara Yonat, with whom I shared the Nobel Prize, had worked with Heinz Gunther Wittmann, who was the director of a Ribosome Institute in Berlin. And they had obtained crystals of the large subunit. So this is the large subunit, and they obtained crystals of that in 1980. And then, a few years later, another group headed by Maria Garber in Russia obtained crystals of the small subunit as well as the entire ribosome. So this was all done around 1980 to 1985 or so. But 15 years later, there was still no good progress on the ribosome. So I decided that after some meetings at which it looked like the Yonat group was stalled, I decided that maybe I should try attacking the small subunit of the ribosome. And this was my lab at the University of Utah. And I persuaded these two graduate students, Bill Clemens, who's now a professor at Caltech, and John McCutcheon, who's now a professor at Arizona State University, that, working on the small subunit of the ribosome, which was still a very large molecule, that it was a good idea for a PhD project. And since they didn't know anything about crystallography or the ribosome, they thought it was a great idea. And this tells you that sometimes it's better not to know anything, because then if you know too much, you realize how difficult it is. If you don't know anything, you just say, okay, sure, I'll give it a try. And so that's what they did. And I like to joke that, see, this is me when I was working in the lab. That's my bicycle, which I still have. And I'm wearing gloves, which shows I was the only one actually doing any lab work when this picture was taken. And I also like to joke that this was in my Bin Laden days, because I had to shave it off after 9/11 because I kept getting stopped at airports all the time. So I thought that, this 30th subunit would be a quiet niche for me because Ada Yonat was working on the large subunit, and the Yale group started to also work on the large subunit. But suddenly, Ada also started to work on the small subunit. In fact, she switched. And so I suddenly found myself in the middle of a four-way race because those Russians who had crystallized the whole ribosome went to work for a very famous ribosome biochemist in California, Harry Noller, who also recruited a superb crystallographer who actually figured out the way to get structures from these large RNA molecules. And my former mentor, Peter Moore, had suddenly become a kind of rival or a competitor of mine. So, so it was suddenly a very tense period. Now, the thing that you shouldn't do when you're in the middle of a race is waste time. And instead, what I did was to move across to a different continent right in the middle of a race. So we did think that was a very foolish thing to do, but luckily it worked out. And the reason I moved is I wanted to work on the whole ribosome structure, but I had no idea how long it would take. And I needed stable funding because, people like Ada Yonat had been working by then for almost 15 or 17 years, and they had still not made the big breakthrough. And so I didn't know how long it would take even for us to get the first preliminary structure. And I also thought, the MRC lab where I work, I'd gone there to learn crystallography. And the reason I'd gone there is they have a lot of experts in the crystallography. So, I thought since this was a very tough problem, if I had experts nearby, then I could get help from them, and get advice from them. it's a little bit like this, you know, if you just want to drive to Adyar, you can do that, you know, it's not a big problem. But let's say you wanted to drive across the Sahara Desert, then you better take along somebody who understands car mechanics and things like that if it breaks down, because you're now pushing the limits of your vehicle, right? So, and then the final thing was that it's an institution with a long tradition of tackling hard problems. They won 2 more Nobel Prizes in the last 5 years, so they're now up to 17 or something Nobel Prizes, including for a new structural technique called cryo-EM. And it's a place where they don't care how many papers you publish, it's more about the quality of the papers you publish. So Fred Sanger, who won 2 Nobel Prizes, only had about 40 papers or so, if you look at it, and an H index of 18, which is not very high, you know. So that's the sort of place it is. So anyway, I then decided in the middle of this race to move from Utah, which is in the western U.S., to Cambridge, England. And you'll notice that, you know, in all these moves, my wife has very patiently moved along with me, sometimes with very young children. And when I decided to move to Cambridge, and I had to accept a 50% pay cut to move to England because of this salary difference between England and Utah. And the cost of living in England was actually much higher than in Utah. So it's quite, financially, quite a big hit. And I told my wife all this, and she said, okay, I'll move if it's important for your work, but I want to tell you one thing. This is the last time I'm going to move. And so even now I get lots of, you know, in the last 20 years, I've had lots of offers from the United States, you know, asking me to come back, often probably with much higher salaries. But every time the conversation only lasts about, you know, 2 seconds, because there's only one word, which is no, you know. And so I've been in Cambridge all along, but I have to say I very much enjoy working there. So anyway, the other thing you heard about from Mr. Stephen about synchrotrons, and you know, synchrotrons are large sources of X-rays, but they're situated in different places. So here we are in Cambridge. We had to go to synchrotrons in France, in the north of England, in Brookhaven, which is near New York, and in Chicago. So I like to joke, and this is what these synchrotrons look like. They're large particle accelerators, which generate very powerful beams of X-rays. And I like to joke that you could join a ribosome lab and see the world. But of course, they only see the world's synchrotrons. They don't actually see them. You know, they're not going to listen to jazz in Chicago or going skiing in Grenoble, because they're working all the time. And essentially, this is what they see. This is a synchrotron instrument. This pipe here brings this X-ray beam, and your crystal is usually held here, and it's kept cold by a stream of very cold nitrogen. And this is the detector that detects the scattered X-rays. So if the experiment all works, then you end up getting a molecular structure, and almost simultaneously, within roughly a month or 2 of each other, the Yale group, and then soon afterwards we, published the structure of the large and small subunit of the ribosome. And these structures immediately answered a particular puzzle, which is, you know, the ribosome itself, as I told you, is made of RNA and proteins. But the ribosome is the machine that makes proteins. So if it itself is made of proteins, how did the ribosome even get started? And the first idea about this was given by Francis Crick a long time ago, in the late 50s, when ribosomes were first discovered. And he said that maybe the first ribosomes didn't have any proteins, they only consisted of RNA. And what we find is that the key parts of the ribosome, the parts that bind mRNA and the parts that stitch together the amino acids, all those critical parts of the ribosome consist entirely of RNA. And the proteins, which are shown in gray here, are mostly on the outside of the ribosome. And so it does suggest that originally the ribosome was an RNA molecule, and gradually proteins were added to itself to perhaps make it more efficient and more, being able to regulate it and so on. And then a few years later, we saw the structure of the entire ribosome. This structure has about 500,000 atoms. So at the time, it was one of the largest structures to be solved by crystallography. But now, using newer methods, people have solved much larger structures. And as soon as the structures of the ribosome came about, we could quickly solve structures of the ribosome with antibiotics bound to it. Now, many antibiotics work by binding to bacterial ribosomes, but they don't bind our ribosomes so well. And so they preferentially inhibit the growth of bacteria or kill them. And so compounds like erythromycin or streptomycin or tetracycline, these are all ribosomal antibiotics. And so we were able to solve these, with whom I had a big argument at the banquet at the end of the meeting. And I found out a few months later that he was appointed to the Nobel Committee for Chemistry. So I thought, well, I have absolutely zero chance of getting the Nobel Prize, and I forgot all about it for 5 years. In fact, after that, I stopped going to Sweden, even if I was invited, because I thought, why should I waste my time? Because I'm not going to be one of the people who gets it. But it shows you the integrity. He was on the committee, but he still voted for me. And in fact, he could have very easily said, well, this Ramakrishnan is a good scientist, but not quite in the same league as this other fellow. He could have easily done something like that, but it shows his integrity. And anyway, I'd given up on the idea of winning, so when I got the phone call, I thought it was some friend of mine trying to play a prank, play a joke on me. So I never would have thought, growing up in Baroda, that I would end up, over here. This is a long central table. There are about, I think, 800 or 1,000 people that are all served simultaneously. That itself is a kind of orchestration. And you can see here, this is the King of Sweden, who's at the center of the center table. And next to him is Ada Jonat, and this is me, and this is the Crown Princess Victoria, and this is Tom Stites. So Stites, I, and Jonat shared the Nobel Prize. So we're next to here. And over here is my wife, Vera, talking to the German vice chancellor, which is to say the vice president. And anyway, this was all fine. Now, of course, the Nobels are given for physics, chemistry, physiology, and medicine, literature, and so on, and economics. But the newspapers realized that year, the really important prize was the ribosome, and not much more than the other prizes. And the reason I say that is because the front page of the newspapers was this. So clearly, they had recognized that the ribosome was important, and it had nothing to do with the fact that this Princess Victoria was sitting in the middle of the photograph. So, anyway, so there we are. And I want to finally say that, look, I talk about this, and I've been awarded a Nobel Prize, but really science is very much a team effort, and these are all the people, starting with those 2 graduate students in Utah who started the project, and Bill Clements actually moved with me to England, and so all these people went through my lab. This is the lab up to 2009, and of course I've had many more people since then. And often they're the ones who come up with ideas, certainly they're the ones who make things work, and so you have to realize that the director of the lab, the principal investigator, is really somebody who has a vision and maybe drives things forward, but could never do it alone. It requires really brilliant people to do it. And so what have I learned from science? So first of all, scientists are neither Spocks. I don't know if Star Trek is, I don't know if Star Trek is popular here, but Spock was this alien who was completely rational. He had no human emotions, okay? So scientists are not Spocks, and we're also not saints, you know? It's not as if scientists are just doing this for completely altruistic reasons. But we're motivated by lots of different things, like curiosity, or ambition, like a desire to succeed and be first. We also have egos and insecurities and anxiety, and we have rivalry and competition. Competition can be bad for scientists, but it can be good for science because it spurs people to work faster and better. And so there's that. And scientists are motivated by self-interest. So if it's in their self-interest to collaborate, they will collaborate. If it's in their self-interest to compete, they will compete. You know, so it's not one thing or the other. You know, it can be both, sometimes both at the same time. They'll be, even with the same person, they'll be collaborating on one project and competing on a different project. But scientists are also altruistic. Humans are also altruistic. You know, for example, my father fell down. He's 97 years old. But 2 years ago, he got dizzy and fainted and fell down in the street and banged his head. And, you know, he was sort of unconscious, but lots of people suddenly gathered and went to help him and then called the ambulance. Somebody called the ambulance and so on. They didn't have to do that. They're not getting anything from them. But it shows you that humans have that altruistic thing. It's what made us successful as a species. And so many people also helped me. For example, many people gave me advice. They provided reagents that I couldn't buy, you know, special reagents that I needed for my work, and they had made them. And they provided them to me free of cost, and they didn't want to be a co-author, you know, just as an altruistic thing to help a fellow scientist. Many of them gave me technical help, you know, how to do certain things, how to collect data from a particular instrument and a synchrotron and so on. And as I said, often with no expectation of anything. In fact, a good friend of mine synthesized a very special compound that was key to solving the structure, and he didn't want to be a co-author on it. So, what about some general lessons? Well, the first thing, if you ask why did my life work out, it's because I kept my options open. So, you know, if I didn't succeed in physics, you know, maybe I could have gotten some, you know, mundane job and forgotten about research, you know. But I thought if I wanted to keep my options open in research, I would change fields, you know, even if it meant going to completely new fields or learning new techniques, like going on sabbatical to learn crystallography. The other is that I've never been afraid to ask for help or for advice, because you know, you shouldn't have what I call false pride. You know, if you don't know something, you should just admit that you don't know it and ask for help. And that's the fastest way to learn something and get advice. The other is you should talk to people, because many ideas I got were often from random conversations with colleagues and so on. So if you simply isolate yourself, it's not going to be that helpful. But of course, you can't spend all your life talking to people. You have to also think hard and, you know, work hard and trying to be original. And another is, everybody has imposter syndrome. So, you know, when I went to the MRC on sabbatical, I thought, you know, these people are all great scientists and I'm just this nobody, and maybe they'll realize I'm a complete idiot, you know. And so you always have this feeling that you fooled people, and they're going to find out one day that you're actually, you know, kind of a fool. And that's called imposter syndrome, and everybody has it. But if you're an outsider, you often feel it even more, because you don't feel that you fit into the club. And this often happens if you're a minority or a woman in science or something like that. And then, of course, science doesn't happen strictly because of your innate talent. You know, it requires a lot of things, and especially experimental science. These are the 4 G's from the German words that Max Perutz used to talk about, which is Gell, Geschick, Geduld, and Glück. Geld means money, you need funding and resources. And skill, you need to be trained well and have the requisite knowledge. And you need patience. You can't just give up at the first sign of difficulty. You need to have patience to sort of keep plugging away at it. And finally, you do need luck. People underestimate the importance of luck. In my case, for example, little random ideas, or even, the luck that Aaron Klug didn't throw away my letter to him and allowed me to do a sabbatical. So there are all kinds of luck that we forget later on. Later on it all becomes, a series of logical steps because of our brilliance, but it isn't actually like that. So I want to conclude finally by showing you a little movie of the ribosome. I hope this works. So, well, I'm sorry the movie didn't work because I had to transfer it to a local computer. So I'll leave it right there. But you can see that movie of the ribosome on YouTube. So if you simply Google my name and ribosome and movie, then you can probably see that movie or translation on YouTube. So I'll leave it there and thank you.